So I have the privilege of introducing our first speaker today, and it's Dr. Thokozeni Lipato. Dr. Lipato is a general internist who has been caring for individuals with sickle cell disease since 2009, who is a primary care physician and has been at VCU Health System since 2013. He has spent most of his career caring for patients who often face significant barriers to adequate health care. In addition to his clinical role, Dr. Lipato is spearheading an effort to open an outpatient infusion clinic for the sickle cell disease program, and he is active in clinical research. Dr. Lipato, thank you so much for joining us, and the floor is all yours.
Okay, so I'm going to give an overview of sickle cell disease. And my goal is to try to put some depth into the conversation about sickle cell disease, but also to make it clear enough that you do not need a medical degree or a biochemistry degree to understand this. So sickle cell disease is a genetic condition that affects the hemoglobin. And the hemoglobin is a protein that is in red cells, and it is responsible for carrying oxygen. And so I'm going to make a little change to these chairs, if everybody can see these chairs. And the change I'm going to make to these chairs to explain sickle cell disease. So the hemoglobin is a protein that has 4 parts, and you can think of it as having 4 chairs. There is what's called the beta chain, which would be these 2, and then you have the alpha chain, that would be these 2. And what is in this protein structure is iron sits here, but also this is where oxygen sits. And so red cells are the oxygen carrying cells of the body because they have hemoglobin, this protein structure. Sickle cell disease is a genetic condition that affects the structure of hemoglobin. So there are different kinds of hemoglobin. In individuals who do not have sickle cell disease, there are 2 kinds of hemoglobins, and they're designated A1 and A2. And the difference is really just what the chair looks like. With A1, you have the alpha chains and the beta chains. In A2, you have the alpha chains and the delta chains. And that's just, it does the exact same thing, looks very much the same unless you're looking under a very high-powered microscope, and it carries oxygen. And then you have fetal hemoglobin. And fetal hemoglobin is the kind of hemoglobin that we all had when we were inside our mother's womb. And again, it is the same structure where you have 2 alpha chains, but instead you have 2 gamma chains. So the chairs just look slightly different. Now, what determines the shape of those chairs is, of course, your genetics. And so, as you know, we have 2 copies of our genetic information from both of the parents. And sickle cell disease, the classic sickle cell disease, is a genetic abnormality that occurs on chromosome 11. And we know exactly where that genetic abnormality exists. So to explain a little bit about why genes and a genetic abnormality results in what a protein structure looks like, we produce proteins based on our genetic information. And the way that occurs is that within our DNA, we have what are called nucleotides. So these are just these nitrogen structures that sit touching each other, and they have a lot of information. They determine almost everything about the human body. And the way that gets translated into a protein structure, such as hemoglobin, is that DNA information gets translated by what's called a messenger RNA. And you probably have heard enough about messenger RNAs with the new COVID vaccines. The messenger RNA is a way that the body takes that genetic information and it translates it so that using that genetic information, it can determine what kind of protein it's going to make. And each segment of that DNA correlates to a certain kind of amino acid. An amino acid are the building blocks of a protein. And so the segments of that DNA corresponds to specific amino acids. And based on what the DNA says, that you will then create a specific protein, like, for example, a hemoglobin. In sickle cell disease, the classic form of sickle cell disease, and I'll get to why I'm saying the classic form, we know exactly where that genetic abnormality is. And we know exactly what happens in the DNA. I said that the DNA is basically these repeating sequences of these nucleotides, these nitrogen bases that spell out a message. With sickle cell disease, there is this genetic abnormality that changes at one point this message. It changes one of those nucleotides. And that one change of that nucleotide within the DNA then gets read by the messenger RNA in a different way, and then it gets translated into a different protein because now it recruits a different kind of amino acid because of that one genetic abnormality. And in the case of sickle cell disease, it results in the recruitment of this one amino acid, which is called valine, when it should be recruiting an amino acid called glutamic acid. And so it's just that one simple change when the body is building that protein molecule that results in sickle cell disease. And so taking a look at the molecule, taking a look at the protein itself, if we can go back to the chairs now, is here we have a sickle cell, a hemoglobin in a sickle cell individual, where now these, if we're saying these are the beta chains, these two beta chains, these two chairs now have a slightly different amino acid that is making up these two particular protein components. So now it looks slightly different. It does the same thing. It has iron, it has oxygen to sit on there, but that chair, or the components of these two chairs, looks slightly different than it does in somebody who does not have sickle cell disease. Now, the reason why I said I use the word classic sickle cell disease is because there are several different variants. And we're going to focus on 4 because these are the variants that are most dominant in the US. There's hemoglobin SS disease. This is what I'm referring to as the classic sickle cell disease because it's the one that was first identified. Then there is hemoglobin SC disease. There is sickle beta 0 thalassemia and sickle beta plus thalassemia. But there are others. There are others with different designations: sickle D, sickle E, and sickle O. And what determines those other variants has to do with where the genetic mutation occurs and how it changes that protein molecule, how it changes one of those two of those four chairs. It changes it in a slightly different way. So, why is that a big issue? Why is it that if the sickle hemoglobin does the exact same thing, it carries oxygen, why does it lead to a problem? The best way to think about this is what we used to learn in medical schools, the oxygen hemoglobin dissociation curve. And what this classic curve tells us, it explains to us the oxygen capacity or the ability of someone's hemoglobin, someone's red cells, to carry oxygen. And so this graph is illustrating that for a certain amount of oxygen in the body designated by pressure, the higher you go, the more oxygen you get in your body, you begin to increase the oxygen saturation into your red cells. So here I am breathing in oxygen, and then my red cells, my hemoglobin in my body gets filled up with the oxygen molecule. The more oxygen I get, the more saturation, the more filled up my red cells are going to get. And there is a very nice curve that you can illustrate. So the solid curve is hemoglobin A, is somebody who does not have sickle cell disease. And the dotted curve is somebody who does have sickle cell disease. And what you notice with this curve is that you need, so let's say if we were trying to reach 100% saturation with somebody who does not have sickle cell disease, you have to get about 60 millimeter of mercury pressure. So that designates how much oxygen you have to get in your body from the air to have that level of saturation. But then when you look at somebody with sickle cell disease, in order to get that level of saturation, you have to get a lot more oxygen into your body. So that tells us that that simple change in the hemoglobin structure drastically affects how well the hemoglobin carries oxygen. And that is one of the classic and central issues with sickle cell disease, is that that simple change drastically changes the oxygen carrying capacity of the hemoglobin in the red cells of individuals with sickle cell disease. Okay, so then that happens. So now you have somebody with sickle cell disease and their hemoglobin, their red cell, doesn't quite carry the oxygen, as much oxygen as somebody who does not have sickle cell disease. Well, what else happens? So let's turn back to these chairs. In the red cell, you have a lot of hemoglobin in one red cell. And these reds, and the hemoglobin is supposed to remain in this 4-chair configuration. So if this whole room was one big red cell, we would have thousands of these 4-chair configurations all around this whole room. And what they should do is they should remain in this configuration all the time. They should be in this configuration whether there is oxygen sitting in the middle or whether there is no oxygen. And so what happens with blood flow is when the blood goes to the lungs, it gets the oxygen from the lungs, and the oxygen sits in the hemoglobin protein structure. And then as the blood goes to all the parts of the body, it drops off that oxygen to the vital tissues. In somebody who does not have sickle cell disease, when this hemoglobin structure drops off the oxygen, it remains in this same structure. However, with sickle hemoglobin, where these 2 chairs have that one little amino acid change, they tend to do something differently. When they drop off their oxygen, they start to stick together. They create what's called a polymer. And so instead of having 4 chairs here and 4 chairs there and just 4 chairs all over the room, they start to coalesce together into this long line of hemoglobin that is stuck together, called a polymer. And so that is something else that happens in sickle cell disease, is that when the oxygen has been offloaded from the hemoglobin then creates this polymer. Well, that causes problems, as you can imagine. One classic problem is that it deforms the shape of the red cell. So now you go from having a round red cell to one that is more sickle-shaped formed. And then on top of that, you can imagine if all of these chairs, this hemoglobin is in this configuration, well, then it's not going to do a very good job of getting oxygen back once it comes back up to the lungs. Another thing that tends to happen is not only are the chairs, or not only is the red cell deformed, but in the blood vessel of individuals with sickle cell disease, that red cell with that deformed, which is deformed by the hemoglobin, those red cells tend to be very sticky. And so they will stick to the inside of the blood wall. So if you can imagine we're in this big room, or here is the blood vessel, and there's all this blood, red cells flowing through, the red cells begin to stick to the inside of the blood vessel. And not only that, but the red cells begin to stick to the other cells in the blood vessel. And one of the most prominent ones are white cells, which fight infection. So now you have the red cells sticking to the white cells, and the white cells are sticking to the inside of the blood wall as well. And that is another important feature of sickle cell disease, is that there is a lot of adhesion going on, that these cells are sticking to each other and sticking to the inside of the blood wall. And so, we can skip this one. What tends to happen is you have one, the red cells are deformed. On top of that, they begin to stick to the inside of the blood wall. That diminishes the blood flow to the body, and it begins to lead to some very classic symptoms that we see in sickle cell disease. So patients will have severe anemia. They'll have a low blood count. And that's because the body is constantly destroying these abnormally shaped red cells. So patients oftentimes have anemia. Sometimes it is very severe. And during a crisis, it can drop even lower. They will have significant fatigue. This is something that is probably poorly appreciated by providers that take care of patients, that because of their anemia, they have chronic fatigue. Severe pain. This is probably the one that we know about and hear about the most, is that patients will have pain crisis because of decreased blood flow to vital tissues. And you can have a pain crisis that comes and goes, or you can have a very prolonged pain crisis, and at sometimes developing chronic pain that never goes away. And of course, because of the severe anemia and the severe pain, many patients require emergency room visits and hospitalizations. Patients also are at risk of getting infections as well. And one of the reasons why patients get infections is because, unfortunately, at an early age, individuals with sickle cell disease will lose the function of their spleen. The spleen is an important organ that helps fight certain kinds of infections. It kind of protects us from them. And because of the abnormally shaped red cells, many individuals with sickle cell disease will lose the function of their spleen, and that increases their risk of having infection. This is a diagram that really shows all of the vital organs that are affected by sickle cell disease. So it isn't just pain. Sickle cell disease is a condition that can lead, and unfortunately oftentimes leads to end organ damage. And it affects, as you can see, the brain, the eyes, heart, lungs, kidneys, the bones. It is really a medical condition that unfortunately is all-encompassing when it comes to the clinical manifestations. So a little bit about the mortality in sickle cell disease. The dotted line is looking at the rate of mortality as individuals go through their life. And the dotted line is around the 1970s. And what we see is that early on in the life of individuals with sickle cell disease, there is a spike in the mortality rate. And this is between 1 and 4. So there was a time when a lot of children with sickle cell disease would die. And then that mortality rate came down, but then it rose up again in their kind of later teenage years. With time, so the solid graph, I believe, is 2009 or 2008. What you see is that initial wave of deaths has been blunted significantly. And that is two things. Goes back to what I talked about with infections, and it also goes to what I did not highlight, but when I talked about other manifestations affecting the brain. Sickle cell disease causes strokes. And unfortunately, with strokes, a lot of the burden is is felt by younger individuals with sickle cell disease, children. And so there was a time when many children with sickle cell disease would die from strokes and they would die from infections. But with a better understanding of how to manage sickle cell disease, the pediatricians, one, vaccinate differently. So they vaccinate children with sickle cell disease differently than they do children without sickle cell disease. That has helped to decrease the risk of infection and decrease the mortality associated with it. And they have also learned how to monitor children with sickle cell disease who may be at higher risk of developing a stroke. And if they find that there are children who are at higher risk of developing a stroke, then there are interventions that can be made to decrease that risk of having a stroke. So those two interventions are responsible for this blunting of that first wave of increased mortality. And then what you then see is that the graph kind of shifts to the right, which means that as healthcare gets better, as treatments get better, the increasing mortality does not begin as early as it used to be. And that, there are many factors that explains that. A lot of it has to do with a lot of the new disease-modifying medications we have, and just a better understanding of sickle cell disease and better funding when it comes to sickle cell care nationwide. But still, unfortunately, sickle cell disease, one of the most important burdens is that individuals with sickle cell disease still have a higher mortality rate. And so if you look at the dark bar is sickle cell disease, then the one in the middle is the African American population as a whole, and then the one after that is the entire US population. And so you can see the increased mortality rate in sickle cell disease compared to the general population and compared to the African American population. And that mortality rate from the beginning is just so much higher in sickle cell disease. So, sickle cell disease is the most common inherited blood condition in the US. We probably don't do a good job of counting how many people have sickle cell disease, but if you read the literature, it's approximately 100,000 in the US. Primarily, it affects African Americans in the US. But you see and get a better understanding for the kind of the global nature of sickle cell disease. So approximately 1 in every 365 African American births will be somebody with sickle cell disease. Within the Latino population, it's approximately 1 in every 16,000 births. Now, that being said, sickle cell disease obviously is a global disease. And this map just shows the prevalence, the worldwide prevalence of where you see the highest incidence of sickle cell disease. And the sickle cell disease, especially the classic one, as I talked about, came about from a genetic abnormality, a genetic mutation. And geneticists believe that that mutation occurred in several different parts of the world, independent of each other. So it kind of explains why those regions in Africa and in Asia is where you have the highest concentration of sickle cell disease, is because that's where the mutation is thought to have first occurred. And then, of course, with the change in humans, particularly driven by the slave trade, you then have sickle cell disease. And I see if I can go back, there we go. You then have sickle cell disease now being seen in South America and obviously in North America. And just, I always like to point out, look at India. There is a lot of sickle cell disease in India. There's a lot of sickle cell disease in the Middle East as well. So it isn't just a condition of people of color, people of African descent, even though that is how we classically think of it in the US. And this just shows kind of where many of these other variants of sickle cell disease, where they tend to be more prominent. And so again, illustrating the global nature of this disease. All right, so talked about the 4 types of sickle cell disease that are most prominent in the US. And I'm going to kind of go through the genetics and kind of how does somebody get sickle cell disease, or how is someone born with sickle cell disease? So sickle cell disease is thought is an autosomal recessive condition, which means that you have to have 2 parents that have the sickle trait. And so, not to confuse you, this is describing the classic sickle cell disease, so hemoglobin SS. So you would have 1 parent with the 2 parents with the trait, and then depending on which chromosome the child happens to get, which is completely roll of the dice, each time a child is born, you can see what your risk is of getting sickle cell disease. So going to the next one, you can see how that risk changes on the carrier state or the disease state of the parents. So if you look at, this would be my top left box, where you have the father has a trait, the mother does not have the trait, then you can see what are the chances of the child either having the trait or not having the trait at all. It's 50-50. So you have this 50% chance of either having the trait or not having the trait. Then if you move over to the top right, where both parents have the trait, there is still a 50% chance of the child having the trait. There's a 25% chance of the child having sickle cell disease, so SS disease, and then there's 25% chance of the child not having the trait or the disease. And then as you go down to the bottom right, if 1 parent has sickle cell disease, 1 parent has the trait, then now you see how the risk changes. That tells us that all of the children are either going to have the trait or the disease. There is no possibility for the child having nothing. And then if you go finally to the bottom left, where you have somebody that has sickle cell disease, so SS disease, and the other parent does not have the trait, does not have the disease, then every single child will have the sickle cell trait. So that just kind of explains how genetics works with autosomal recessive. So what about hemoglobin SC? So that was SS. That's kind of the classic kind of sickle cell disease. SC is simply, and we can go back to these chairs. Hemoglobin C is again a genetic abnormality or mutation that changes something about the beta chain. So with classic sickle cell, SS, it added 2 valines to these chairs, and it slightly changed the structure. Where with hemoglobin C, it simply adds 2 lysines. So it's just a different genetic mutation in the DNA that results in the beta chains, those 2 chairs, just looking slightly differently. So that's what hemoglobin C is. And you can have people with hemoglobin C disease, where all of their chains are affected like that. Or you can have somebody who just has hemoglobin C as a trait. So this now is the way, if I were to have hemoglobin SC, so that's the kind of sickle cell disease I have, this is the way the pattern would work, where you would have one parent who has the sickle trait, you would have the other parent that has the C trait, and then they have children, and you can see the risk. And you can change this picture where you have one parent has SS, so they have sickle cell disease, and one parent just has the trait. You can map it out and see again what the risk is. Now, the other 2 sickle cell diseases or variants, sickle beta thalassemia. There's sickle beta 0 and sickle beta plus. Understanding beta thalassemia, it is a genetic blood disorder all of its own. So, to go back to these chairs, one thing to understand about it, there are many, many different kinds of genetic mutations that can result in beta thalassemia. And they have varying degrees of impact on the hemoglobin, on these chairs. So one way to think, one variant of beta thalassemia is where you have a genetic mutation that simply makes you lose one of the beta chains. You may have another genetic abnormality that will make you lose the other beta chain. So now you are looking at a hemoglobin molecule that has fewer chains. That is a very simple way to explain it, because if I get more in depth in it, I'll confuse myself, because the genetics is so complicated that there are varying degrees of severity. So you have some people who have beta thalassemia that is very mild, that they wouldn't even know that they have this genetic abnormality. And then you have some individuals who are born with beta thalassemia that may die at a very young age, and for life-sustaining therapy, they need chronic transfusions from the beginning. But it has to do with how many beta chains you have and whether the genetic abnormality makes you lose some of those chains or all of those chains. So, what is sickle beta thalassemia? It is where you have one parent who has the sickle trait, just like all the other parents, and then you have one parent that has the beta thalassemia trait. But you have to know what kind of beta thalassemia trait that parent has. because they may have a trait that results in more of those beta chains to be gone or less of them to be gone. So there is a variant in sickle cell disease called sickle beta plus thalassemia. And again, you can map out the risk of a child being born with that really the same way we did all of the other examples, looking at sickle cell trait and beta plus or beta plus trait. But if you get, so with sickle beta plus thalassemia, it tends to be a variant of sickle cell disease that is less mild, or sorry, is milder than either sickle cell disease, the classic one, SS. And so many patients will have either a completely normal hemoglobin or they will have a slight anemia, and they tend to not have a lot of the manifestations of hemoglobin SS. So less sickle cell crisis, less anemia, less pain. I say that they tend to because not every human being reads textbooks and follow what the textbooks write. But generally, when we see somebody who has sickle beta plus thalassemia, that their manifestation tends to be mild. And then, if you have two individuals, one has a sickle cell trait, one has the thalassemia trait, but it happens to be the kind of thalassemia that leads to more of those beta chains missing. When they have children, then there's a chance that one of those children will get sickle beta 0 thalassemia. And this kind of sickle cell disease, this variant, tends to be as significant when it comes to clinical symptoms as the classic one, as the hemoglobin SS. So they tend to have much lower hemoglobins, and they tend to have just as many sickle cell crises as somebody with the classic form of sickle cell disease. And so when we see these individuals in our clinic, what we tend to see that individuals with hemoglobin SS disease, the classic form, and individuals with sickle beta 0 thalassemia tend to be the ones who have more sickle cell disease complications. And the people with sickle beta plus thalassemia tend to have the least sickle cell manifestations and complications. And then individuals with hemoglobin SC tend to fall in the middle of that range. But like I said, not every human being reads textbooks, so they don't always fall into those categories. But that is what we generally see in our clinic. So quickly, this just gives it a nice understanding of where sickle cell disease, how it compares with another very classic, or I should say another chronic medical condition, cystic fibrosis. This is a good comparison because it illustrates really the power of funding and research when it comes to making a change in a disease. So cystic fibrosis is a condition that primarily affects Caucasians in America. And that is I think that's blue line, and you can see sickle cell disease is the yellow or orange line. And what you immediately see is that sickle cell disease was seen as a clinical manifestation by doctors much early on compared to cystic fibrosis. The genetics of sickle cell disease was understood much faster or sooner than cystic fibrosis. But then when you start to look at the funding, the development of cystic fibrosis centers in the US, you see that those milestones, those accomplishments when it comes to funding cystic fibrosis happened a lot faster than with sickle cell disease. It took a longer time before we started having sickle cell centers in the US. And that really talks to the disparity when it comes to the management of sickle cell disease compared to other genetic conditions in the US. And this too just shows publications and drug funding, that here you have cystic fibrosis, again in blue, the genetics having been discovered decades after sickle cell disease. Look at the number of disease-modifying medications for cystic fibrosis. Compare that with sickle cell disease. It took a long time for there to be a disease-modifying medication for sickle cell disease. Again, it speaks to the disparity when it comes to funding and research. And that is why funding and research for any disease, especially diseases that are rare, is very important. And it takes a lot of advocacy work, both on the local level, but the national level. I think Dr. Smith is probably going to talk more about this at a later talk. And that largely ends my slide. I won't go over this, so. All right, I will open it up to any questions, and I might need a microphone. Let's see. Oh, someone's giving me a question, is that true? Okay, is that nice? All right, thank you. How prevalent are the... Is that a question for me, or is that somebody, for me to ask them, or okay. How prevalent are the SC variants? Okay, that's a good question. I would say, looking at our population, in Richmond, I don't have, this is just going based on my clinical experience, is that SS is the most prevalent, but we actually see a fair bit of hemoglobin SC in our clinic in Richmond. And I would say I probably see it, I'm surprised at how prevalent it is. I feel like we see that SS is the most prevalent in our clinic. Then I'd probably say SC, or maybe, and then I would say, it's hard to figure out between SS and sickle beta 0 thalassemia, because unless you see them as children and you have a good understanding of the genetics, when they come to the adult side, they clinically look the same. But I would, if I were to guess, I would say in our clinic, SS, SC second, maybe sickle beta 0 third, then sickle beta plus fourth. And I would gladly let Dr. Smith correct me when he comes to talk about something else. But that's what I would say. But to answer your question more directly, it is actually, surprisingly, we see a lot of it in our Richmond clinic. It is not a rare variant. It tends to be more common, but it's not the most common one. SS is the most common one. Do those with only the sickle cell trait, so do people with sickle cell trait present with sickle cell disease symptoms? That's a good question. Most people with sickle cell trait have no symptoms at all. No anemia, no pain. But there are times and there are cases where individuals with sickle cell trait will have symptoms. There are cases of people with sickle cell trait having a sickle cell crisis, manifesting as a crisis under extreme moments of extreme physical stress, whether it's sports or in the military. There have been cases of that. We've seen patients in our clinic that get referred to us about having chronic pain and wondering if it is related to their sickle cell trait. And it's very hard to say yes or no, but by and large, sickle cell trait is not a condition that has symptoms that most people would recognize or need to be treated. Would you be able to explain the chair logic? Had lost the audio and missed that part. Okay. Probably want to, okay, perfect. So, what I was saying about the chairs, and basically the whole reason why they're here, is to explain what the hemoglobin is. So the hemoglobin is the protein in the red cell that carries oxygen. And the protein has 4 components. So these 2 chairs look alike. These 2 chairs look alike. They're slightly, these 2 are slightly different than those 2. These 2 chairs would represent the beta chain. These 2 chairs here would represent the alpha chain. And then the hemoglobin sits in the middle. And so I showed these chairs so that you have this kind of visual understanding of what the hemoglobin protein in the red cell looks like. And when you have sickle cell disease, it is literally a genetic abnormality that changes slightly the structure of this chair and changes slightly the structure of this chair. And that change in the structure of these two beta chairs changes the oxygen capacity, carrying capacity of the entire structure. And I use these chairs to also explain thalassemia, which is a completely different genetic blood disorder, where with thalassemia, you're not changing the structure of these two beta chairs. You are simply changing whether you actually have two beta chairs. And so now you have this hemoglobin molecule that is without one of these chairs. And if it's significant enough, you can actually lose both of your beta chairs. So now you have a hemoglobin that simply has two alpha chairs and it's lost its beta chairs. So that's how I was using those chairs to explain sickle cell disease. How many non-African Americans and Hispanic Americans do you see with sickle cell? Okay, so I would say that in our Richmond clinic, I personally have not seen any patient who I would not identify as African American. Maybe Dr. Smith has seen some. I used to work in Minneapolis. And in Minneapolis, yes, I took care of, well, let me rephrase that. We do have Africans in our clinic in Richmond. In Minneapolis, I did take care of patients of Latino descent, and I had two patients. One was from Saudi Arabia, and the other patient was from Yemen. So I think it is probably just where we are as a location. I think if you spoke to the big sickle cell clinic in Detroit, they probably would have a lot more patients of Middle Eastern descent. And if you spoke to a sickle cell clinic in California, they would have a lot more patients with Latino descent. With the difference between the sickle beta 0 and sickle beta plus thalassemia, there definitely is a difference in severity. And I think that the genetic reasoning has to do with the thalassemia component, that with the sickle beta plus thalassemia, there is less loss of the beta chain. With the sickle beta 0 thalassemia, there is more profound loss of those chains. So I think that explains the difference, why sickle beta plus tends to be milder than sickle beta 0. With hemoglobin SC, so when I try to think about hemoglobin SC with SS, and I'll probably default to my smarter colleague, Dr. Smith, I honestly don't know. What I will say is we tend to see two things occur a lot more as far as early on and in frequency or in prevalence in sickle so SC compared to SS, we tend to see more sickle cell retinopathy, so where the sickle cell disease is affecting the eyes. And in our clinic, we tend to see sickle cell disease in SC impact the joints, so causing avascular necrosis in your hips earlier than we see in SS. You get all of those same complications in SS. But those are the two things that I tend to see earlier in individuals with SC than with SS. I have actually looked to see if I could find an explanation. I have not. Okay. They wanted me to read out so the question was, have you seen a noted difference in disease severity? If so, what do you think is the underlying etiology genetics? So that's what I was answering. All right. Oh, okay. Yeah, I kind of didn't, kind of glossed over that. The question was, can I explain fetal hemoglobin? Okay, yes I can. So, back to the chairs. Fetal hemoglobin is, can you please discuss fetal hemoglobin, hemoglobin presence. So, fetal hemoglobin is again a hemoglobin protein. It has 2 alpha chains, just like adult hemoglobin, I should say. But it has 2 gamma chains. So these are no longer beta, these are gamma. And so it's basically just the gene determines the structure of this protein. And so it recruits a different amino acid, creating these chairs to be gamma, and these chairs are still alpha. It does the same thing. It carries oxygen. But what's interesting, going back to the oxygen carrying capacity, is that fetal hemoglobin binds to oxygen very strongly, stronger than normal hemoglobin, or I should say adult hemoglobin. And the way I explain this to my patients is I say to them, when we were in our mother's womb, we had, the mother has no conscious way of sharing oxygen with the baby. So the baby just has to get the oxygen from the blood as it circulates. And so because of that, because the fetus has fetal hemoglobin, their hemoglobin has the ability to grab onto the oxygen with a higher, stronger ability than adult hemoglobin, and therefore is able to survive in that environment where the mother isn't consciously siphoning off oxygen to the baby. After we're born, in our genetic makeup, for most of us, the gene that produces these 2 gamma chairs switches off, and we stop making this kind of hemoglobin. So we stop making these gamma chains. In somebody who does not have sickle cell disease, you start making the beta chains with the glutamic acid, that normal amino acid there. With somebody who does have sickle cell disease, they lose their fetal hemoglobin, and now they begin making their beta chains that has that abnormality there. Now, that's the simple explanation for hemoglobin F. There are also some implications of hemoglobin F when it comes to beta thalassemia, that some people, certain kinds of beta thalassemia or sickle cell beta thalassemia, you actually have some of that hemoglobin F. So it isn't a all or nothing. You can actually find some people who don't turn off all of their hemoglobin F. And so we have people walking around who still have hemoglobin F in their body. If I have hemoglobin F, which I don't because I've checked it, it would really probably benefit me none whatsoever. But for somebody with sickle cell disease, if they happen not to turn off all of their hemoglobin F, meaning that some of their hemoglobin has the F still there, then that actually becomes beneficial for sickle cell disease because it helps to ameliorate many of the downstream sickle cell manifestations. And I'm going to talk more about the hemoglobin F in the next talk when I talk about treatments for sickle cell disease, because yes, hemoglobin F factors importantly in treatment for sickle cell disease. Last question. With sickle cell disease, do they produce any normal hemoglobin or all hemoglobin sick? Okay, good question. So it depends on the variant. Let me try to think about all of the labs I see. Individuals with hemoglobin SC tend to have no normal hemoglobin. And what's, again, is normal hemoglobin? A, or A1, and A2. And so when I think about the labs that I get, I tend to see people with sickle SC who have, it's almost 50-50. All S, half C, and maybe a little bit of A2. I don't think I see any A1. A1 by far is the majority. So in me, I don't have sickle cell disease. So I probably have 90-something% of A1, then some A2. With SC, it tends to be very little A2 and half S and C. With SS, I don't see any patients with SS having A1. They have a lot of S. It can be 80%, 90%, or even greater of just S, and maybe a small amount of A2. With sickle beta thalassemia, it depends. If it's sickle beta plus thalassemia, then they will have a little bit of A1. In fact, that's one of the things that helps us determine whether it's sickle beta plus thalassemia, because we see a nice amount of A1. I think sometimes I can see it as maybe 10%, 12. If it's sickle beta 0, we don't see any A1. We can see some A2. What's interesting with the thalassemias is we do start to see a little bit of F in there. And that's another clue when we're looking at the labs that helps us figure out which variant it is. Okay, I think we're done with questions. So thank you very much.